Hi, I continue the lecture about hemoglobin. As you know, the hemoglobin has two genes, alpha and beta. In the fetus life, the hemoglobin called hemoglobin F or fetal hemoglobin. I explained how the structure of hemoglobin changes in this trimester. In the first trimester, alpha gene produced the zeta subunit instead of the alpha subunit. Okay? But after first month, the fetal start produce alpha gene in the higher equal amount of the zeta subunit. And after first trimester, the alpha subunit will be produced, but zeta subunit will be stopped. Then in the first trimester, the hemoglobin has two zeta subunit, okay? And in the beta gene also, in the first trimester produced the epsilon subunit, and also start produce gamma subunit. Okay, in the first trimester, the epsilon subunit production will be decreased, but in the first trimester, mainly we have in the high amount of the zeta 2 and the epsilon 2 hemoglobin. Okay, after the first trimester, the second one we will have in the beta gene just to reduce the gamma subunit. Okay, and alpha gene also produce mainly alpha subunit. This one we call hemoglobin F or fetal hemoglobin. Okay, afterwards, mainly the gamma subunit will be decreased in production and the beta will be increased. Okay, as you know, the hemoglobin F has a very short half-life, and the life of the hemoglobin is very short. Okay, compared to the normal hemoglobin. In this case, as you know, after birth, the amount of the degradation of the hemoglobin F will be increased. And in this case, we can see the yellow color under the skin of the fetus, in the baby, and mainly we use the special lamp for the conversion of this unconjugated bilirubin under the skin to the conjugated bilirubin, and it can easily eliminate by the urine. Okay, and it is a normal situation. And the second one, hemoglobin 1AC, this hemoglobin normally can bind to the glucose, and in the diabetic patient, we can, with the amount of the hemoglobin 1AC, we can calculate how was the diet of the patient in the last 3 months. Because, for example, if the patient hasn't diet and use high amount of glucose in the first months and then later has a diet restrictedly, in this case the amount of the glucose in the blood decreased, but hemoglobin A1C never be decreased. In this case, mainly the clinics has both of this for the diabetic patient, because it shows how was the control of the sugar in the blood during the 3 months, and it is very, very important, because this one is easily combined to the hexose or glucose sugar. I continue with the type of the disease. Hemoglobin A is a normal hemoglobin. We have alpha beta subunit. We can have a different disorders, SC or CS. The first one, hemoglobin S or sickle cell hemoglobin, which, as you know, the normal red blood cell is like this shape, because it can easily pass from the capillary, and we don't have any problem. But in the sickle cell anemia or S hemoglobin, the shape will be like this form. As you see here, the shape will be this, and it cannot easily pass from the capillary, and mainly sedimentation rate is high, affinity to oxygen very low, and this type of hemoglobin is very big. What is the problem? It is in the beta gene. Why it is in the beta gene? Because in the amino acid number 6, which is important, there is an exchange between the amino acid glutamic acid to the valine. Okay, in the glutamic acid change to the valine amino acid in the position 6. Okay? In this case, in the beta globin, the shape of the will be different, like a moon shape, and it will be it has a very low affinity. In the hemoglobin C, it can be caused mainly because of the mutation. Mutation, but usually can be because of the point mutation, because in the point mutation, as you know in the biology, just one amino acid can be changed. And in the hemoglobin C, also can be because of the point mutation. In the also beta subunit, glutamic acid changed to the lysine. Also, glutamic acid change to the lysine. Okay, it is also has a low affinity. Also, the problem is that it has a high hemolytic anemia, because the hemoglobin C, mainly patient blood start to degradation and the hemolysis happen more. The life of the hemoglobin is short. Sometimes both of them happen, and we can call it SC, hemoglobin SC. This disease can be because of the both mutation. Normally, sometimes it is without symptomatic, but usually because of the high sedimentation, accumulation of the blood vessel, and also in the small vessel and some gland, for example, pancreatitis happen, and it is very strong disease. It is all of them. It is because of the point mutation or other mutation in the beta subunit. And other problem we can say it is a thalassemia. It is a special disease. It can be because of the change to the alpha and beta thalassemia, and it can cause anemia, and it is very important disease. And it is because of the genome problem. Mainly, it is because heredity can be happen main cause of this disease. Okay. Today we understand about the life of the hemoglobin. It is usually 120 days, but the other hemoglobins, fetus hemoglobin F, and also hemoglobin C and hemoglobin CS has a short life. And we understand about they don't have a mitochondria, mainly use the glucose as a energy source, as a glycolysis, and because of this, the hemoglobin 1AC is very important for the control of the glucose in the long time in the diabetic patient, and this is very important. And also during a fetus, which hemoglobin can be produced and how they can change, and all of them belong to the globular protein and the subfamily of the heme globular protein, and which is important. I hope the second lecture was good and useful. In the next lecture, I will speak about fibrillar protein, and then I will go to the enzymes, and I will speak about the enzyme. I hope it will be good. Have a good time, and see you in the next lecture.